Okay, in this video, we're going to be talking about protein structure and function. So far, we've talked about how to take an amino acid and polymerize it into an oligopeptide. And if I were to create an analogy, that's like creating a red piece of paper. And in the cell, that single polymer isn't functional yet. It has to be folded into this unique shape. In this example, a paper crane, which then has a function in the cell. And so the topics for this video are going to be to list and describe the 4 levels of protein structure. How does it get to that 3-dimensional shape? And then understand that structure is related to function. So what do proteins look like? Proteins have unparalleled diversity in their size, shape, and chemical properties. And so you could have a protein like porin, in which each individual amino acid is depicted by a green circle, and it creates this essentially donut-shaped protein. And what porin does is it's actually a pore, and so things can pass through this hole and access places in the cell they wouldn't normally be able to get to. And so here, structure is really clearly related to function. Here, there's another protein. This is an enzyme. It's called chymotrypsin. This is an enzyme that hydrolyzes certain amino acids from a polypeptide. And so here's a target protein, and this might bind to the active site. But you can see of the enzyme, which we'll talk about in the next class. But what you can see is that only a certain part of the protein interacts with its target. And so we can zoom in and look at what amino acids are there that might give this enzyme that activity. Okay, so let's talk about how these proteins get organized into these 3-dimensional structures. So all proteins have just 4 basic structures: primary, secondary, tertiary, and quaternary. And let's talk about what each of those are. So when I talk about them, pay attention to a couple things. The bond or types of bonds and interactions that characterize that structure, and then what that structure ultimately looks like. Okay, so the primary structure is characterized by peptide bonds. So essentially, you've already learned about the primary structure. And so here's a really short oligopeptide. It's just made up of 8 amino acids, and each of these amino acids is joined by a peptide bond. And this is the primary structure, just the linear primary structure of this oligopeptide. So it's very simple, but it's also fundamental to all higher levels of protein structure. So the primary structure will affect secondary, tertiary, and quaternary. And because of this, if we get a single amino acid change, this can radically alter the protein shape and therefore the protein function. And so as an example of that, let's take a look at hemoglobin. So if we were to zoom in on the structure or the sequence, the primary sequence of hemoglobin, we would see that amino acid 5 would be proline, followed by glutamic acid in position 6. And hemoglobin is one of the proteins that carries oxygen in red blood cells. And so here's what a picture of a normal, healthy red blood cell looks like, where it has this concave-like structure. Essentially, it looks like you put your head in a pillow. But people who have sickle cell anemia, their hemoglobin has a mutation that converts the amino acid at position 6 to valine. And this affects both the ability of this red blood cell to carry oxygen, as well as the overall shape of this red blood cell. And the shape of red blood cell doesn't flow as easily, and this can cause problems and complications that people experience with sickle cell anemia. So this just highlights how fundamental that primary structure is. Moving on from the primary structure is the secondary structure. And the secondary structure is characterized by hydrogen bonds. So again, paying attention to those bonds. This one's characterized by hydrogen bonds, but specifically hydrogen bonds in the backbone, in that peptide bonded backbone. Okay, so it's between a carbonyl group of one amino acid and an amino group of another amino acid. So let's just take a deeper look at this. So here's a carbonyl group, and oxygen is more electronegative than carbon. And so the electrons in this double bond are going to spend more time orbiting around oxygen than they are carbon because it's more electronegative. And so this is going to give the oxygen a partial negative charge. Now, nitrogen is also a little bit electronegative, especially relative to hydrogen. And so here we're going to have the electrons spending more time around nitrogen than hydrogen, and this is going to give this hydrogen a partial positive charge, okay? And so because we have a negative charge and a partial charge on this oxygen, we can have a hydrogen bond form, okay? Just like in water, except, right, this is in the same molecule now, or the same polymer. Okay, so this is secondary structure. Again, hydrogen bonds in the backbone. The secondary structure is also where the protein starts to fold, because these hydrogen bonds can only occur when the polypeptide bends so that the carbonyl and the amino group are close together. And this can result in two shapes. So the first shape I'll talk about is an alpha helices. So in this example here, we have the polypeptide essentially forming a spiral. So if you were to take a pipe cleaner and wrap it around your finger, that's how a alpha helices would look. And in this example, every 3.4 amino acids, we have an amino group forming a hydrogen bond with a carbonyl group as we go down this spiral. And scientists often represent these alpha helices in what are called ribbon diagrams, and the alpha helices is always shown as kind of this spiraled ribbon-like shape. The other structure that can form as a secondary structure is a beta-pleated sheet. And a beta-pleated sheet forms when the protein travels in one direction. Let's have it go this direction. And then a kink forms about turning the protein 180 degrees, and then the protein travels in the other direction. And hydrogen bonds can form between these parallel parts of the protein. And so they can form opposite each other. And in a ribbon diagram, these are often represented by these arrows where the protein is kind of traveling in one direction. Then we have that turn, and then the protein travels in the other direction. And these, again, can be this first form, or this first time the protein begins to take shape. And this happens because of hydrogen bonds in the backbone. And while a single hydrogen bond may be weak, over the entire length of the protein, because of the large number of hydrogen bonds, this gives the protein a lot of stability. So the next structure we'll talk about is the tertiary structure. The tertiary structure is the distinctive overall three-dimensional shape of the protein, and it involves bonds and interactions between R groups or between R groups and the backbone. So again, just to compare this and contrast this to secondary structure only involves bonds with the backbone. So the bonds in tertiary structure include covalent disulfide bonds, ionic bonds, hydrogen bonds, hydrophobic interactions, and van der Waals interactions. And for your notes, I've written down what those all are, but I thought it might be more useful to take a look at what those look like in a ribbon diagram of a protein. So here's a ribbon diagram of a protein. Here's the alpha helices formed by hydrogen bonds between the carbonyl group and the amino group in the protein backbone. But forming the three-dimensional structure, we have hydrogen bonds, and hydrogen bonds can occur between a side chain. So here we have a side chain with a hydroxyl group coming out, and that hydroxyl group, because the oxygen is going to pull the electrons, it's going to create a partial positive charge on this hydrogen. And then this carbonyl group, this is part of the backbone, and the oxygen is more electronegative, so it's going to have a partial negative charge. And so here we have a hydrogen bond forming between a side chain and the backbone. We can also have a hydrogen bond forming between two side chains with partial charges. This nitrogen would have a partial negative charge, and this hydrogen would have a partial positive charge because it's bound to oxygen. So we would have a lot of hydrogen bonds, which would build in strength over the whole length of the polymer, or over the whole length of the oligopeptide. Here are the disulfide bonds forming between two cysteine amino acids, and those have those sulfohydryl groups. This is the strongest of the bonds within the tertiary structure because it's a covalent bond. The ionic bond is also a strong bond and occurs between two side chains that have opposing full charges. So here we have a side chain that is acidic, and here we have a side chain that's basic, and so we have these full charges, and so an ionic bond can form between them. The last interactions to talk about that help form the tertiary structure are the hydrophobic interactions and van der Waals interactions. So there are amino acids that are primarily made up of carbon and hydrogen, and they want to avoid interaction with water. And so to do this, what they do is they cluster within the three-dimensional structure of the protein. And so they're packed in pretty tight, so they're hidden away from water. And because they're packed in pretty tight, they're more likely to interact via van der Waals interactions. So just to remind you what van der Waals interactions are, it's when the orbit of electrons around an atom, around the atomic nucleus, is asymmetrical, and it creates partial charges in that in that atom. And so that atom can interact with other atoms with that with an opposing partial charge, and form a tighter interaction that stabilizes the protein. So lots of bond types, pretty much all the bond types involved in the tertiary structure, and R groups play a very important role. Tertiary structures are very diverse. And so you can have tertiary structures composed mostly of alpha helices, like this protein here. You can have tertiary structures composed mostly of beta-pleated sheets. And you can have tertiary structures that are a mix of everything. So we have this protein has 1, 2, 3 disulfide bonds. It's got an alpha helices, a beta-pleated sheet, and then it has some parts of the protein with no secondary structure at all. But these are all stabilized by these interactions with the R groups of the amino acids. The last type of structure to talk about is the quaternary structure. And this is the bonding of two or more distinct polypeptide subunits. So not all proteins form a quaternary structure, but some do. And so an example of a protein that forms a quaternary structure is this CRO protein from a bacteriophage. And one polypeptide interacts with another polypeptide using the bonds and interactions that we just talked about for the tertiary structure. It's just these are two distinct polypeptides. And they're only functional once they come together in the quaternary structure. Another example of a quaternary structure is the protein that we talked about at the beginning, which is hemoglobin. Hemoglobin is made up of 2 alpha chains and 2 beta chains, and these distinct polypeptides, these 4 polypeptides, come together in a tetramer, and they're actually held together by ionic bonds. And they're only functional once all 4 of the polypeptides come together. So these are relatively simple, but some cells contain molecular machines with lots of polypeptides that compose one central complex, and that complex carries out one particular function. So the evidence demonstrating that protein folding was required for protein function came from a scientist named Christian Anfinsen. And he was studying a protein called ribonuclease. And this protein is shown here, and it has a bunch of disulfide bonds, and it has alpha helices held together by hydrogen bonds. And when this protein was folded or isolated in a solution, it was functional. And Christian Anfinsen then added this chemical that's called a denaturant. And a denaturant will unfold or denature a protein. And so what happened was when this chemical was added, all the disulfide bonds, those disulfide covalent bonds, were broken apart, and all the hydrogen bonds were broken apart. And when Christian tested whether this protein still had activity, he found that it did not. But he was able to remove the denaturant, and the protein spontaneously folded back and was functional again. And so this was the first evidence really clearly demonstrating that protein folding was required for protein function. Although some proteins can fold up spontaneously, they might not do so correctly. And so that's why molecular chaperones are really important to help with protein folding inside the cell. And so here's an example of a protein where the different colors represent different amino acids. And you might have this stretch of amino acids that are non-polar, and so they want to avoid an interaction with water. But if these were just kind of made and interact with water, we might get them interacting with multiple proteins at the same time to kind of hide those hydrophobic stretches. And then you'd get this non-functional aggregate. But instead, in the cell, these other proteins, which are called molecular chaperones, come along and they bind to and sequester those non-polar regions, allowing the protein to eventually fold and take the correct shape. And once they're correctly folded, the molecular chaperones leave those proteins, and these guys can go out into the cell and do their important jobs. What are those jobs? So proteins play a very important role in catalysis or enzymatic function, and so helping out chemical reactions. And so here's an example of a protein enzyme. And protein enzymes have this really important area called the active site. And in the active site, there's usually some amino acids, or we call these key residues, that can act like molecular scissors to clip off a part of their substrate, or even help kind of bring together two different substrates depending on what the enzyme does. We'll talk more about enzymes specifically next class. Proteins can also play a really important role in structure, like in your hair, which is in your nails. Your nails are made of keratin. In movement, so we'll talk a lot about the actin cytoskeleton, which helps cells crawl. Cells play an important role in signaling. We'll talk about the function of insulin and how that helps signal for the release of glucose into the blood. Proteins play a really important role in transport, so bringing cargo through neurons, for example, and in defense. So some of my favorite proteins are antibodies. They're actually quaternary structures, and they help defend our body against pathogens. So proteins are super cool. They're probably my favorite macromolecule. So I just want to end this video by reviewing the types of protein structure. So we have the primary sequence of an amino acids that makes up the primary level of protein structure, and it's characterized by peptide bonds. Moving on, we have hydrogen bonds in the backbone that form either alpha helices, the spiral, or those beta pleated sheets, which have turns. Bonds between R groups, all kinds of bonds between R groups, or R groups in the backbone help the protein take on its three-dimensional shape, and that's the tertiary structure. And then finally, some proteins are produced by combinations of polypeptides, again, held together with bonds between R groups or R groups and the backbone, and that's the quaternary structure. So that's it for this video, and I look forward to seeing everyone in class. Thanks, bye.